Hello everyone. In this set of lecture, we will be talking about different classifications of parasympathetic nervous system. So, as we know, parasympathetic nervous system is broadly classified into two basic categories. So, if we talk about parasympathetic or cholinergic nervous system, then we can basically classify it into two types. One is the nicotinic nervous system, and the other is the muscarinic. Right? Both of them has different type of receptors involved, and they play different roles. So uh, just to give you a brief overview of what happens in these type of classifications. Basically, nicotinic receptors or the nicotinic innervations, those are mainly ion channels. Okay, so the nicotinic receptors are basically ion channel in nature, whereas the muscarinic receptors, these are basically G protein coupled receptors. I'll be talking about them in details when I will discuss about them one by one. Okay, but for the time being, just keep in mind that the nicotinic receptors are basically ion channels in nature. They are basically pentameric in structure, whereas the muscarinic receptors are GPCR in nature. There are different type of muscarinic receptors present as well, based on the location, based on their uh, type of GPCR activity. Okay, so first of all, we'll be talking about nicotinic receptors. Okay, okay. So as I already told you that nicotinic receptors are basically ion channels in nature. So these are basically pentameric ion channels. So what does pentameric means? Basically, there has five different subunits that combines together to form a complete ion channel. Okay, so if I have to draw a basic structure of nicotinic acetylcholine receptor, or in short, we can also write it as nicotinic acetylcholine receptor. Okay, so basically we can draw a diagram of a receptor. This is a very rough diagram. Okay, just to make you explain uh, what does pentameric means. Okay, so suppose this is a type of ion channel and it has five different subunits. Suppose, yeah, 1 2 3 4 5, like this here as well. Okay, so it has five different subunits, and five different subunit combines together to form a complete ion channel, and these ion channels are present transmembrane to the cell membrane, right? So this is the cell membrane, this is the extracellular region, right? And this is the intracellular region. Okay, and this is obviously the transmembrane region. Now what happens is, when these ion channels are activated, they basically causes both influx as well as efflux of certain ions. But the prominent one, the most prominent one, is the influx of sodium ions. Okay, so whenever these ion channels are activated, they causes influx of sodium ions. Apart from sodium ion, it can cause influx of calcium ions as well. Okay, so basically they you can say they are excitatory in nature, because they are causing influx of cations, and we know that if influx of cations are taking place inside the cell, that can cause depolarization of the cell, and depolarization can further increase the action potential. uh, it can cause muscle contractions, and many other factors. Okay, now, apart from these sodium and calcium ions, it can also cause efflux of another type of ion. Okay, though this is also a cation, this is basically a potassium ion. Okay, but this the prevalence of efflux of potassium ion is very less as compared to influx of sodium and calcium ions. Okay, but just for the information, you have to know that it can cause influx of some cations, as well as to some extent, to a smaller extent, efflux of certain ions as well. Okay, so, if I have to draw the structure of this nicotinic receptor in bit more details, I can draw it like this. So, as I told you, it is a pentameric structure. So, suppose this is one subunit, this is another one, this is the third one, and at the back we have two more subunit present. Okay, so and this is in the transmembrane region. Okay, and similarly, this is the pore region. Okay, this is the pore region present just behind this front subunit. Okay, so whatever ion is coming, it causes influx of basically sodium or calcium ion, as well as it can cause efflux of certain potassium ions from the cell. So you can refer to any of the two structures. Okay, both determines the same ion channel, but for the simplicity I will from now mention this structure of nicotinic receptor. Okay, I think this is a bit easier to draw, and I can explain bit easily, properly, in a clear way in this particular structure of nicotinic receptors. Okay, so basically what are these different subunits? Okay, so if we see uh, the subunits, like as I told you, there are 5 different subunits, right? So what are these subunits basically, and what are these known as, right? So basically this nicotinic acetylcholine receptor forms alpha 2 epsilon subunits. So we can have alpha, beta, gamma, delta, or epsilon subunits, right? Now they are arranged different in a different ways, in a different combination, to form a complete nicotinic receptors. Okay, so before discussing more about their arrangement, I would like to tell you that nicotinic receptors are basically of two types. Okay, so uh, nicotinic, or we can call it as nicotinic acetylcholine receptors, these are of basically two types. I have discussed about them in the first video lecture, in the introduction portion of autonomic nervous system, about this as well. But in details, I will talk about the classification of nicotinic receptors here. So basically, nicotinic acetylcholine receptors are divided into NM as well as NN receptors. So what does M stands for? It is basically muscles. Okay, so we can also call it as muscle types, or nicotinic muscle type acetylcholine receptors, right? So this is the muscle type, whereas N stands for neuronal. So this is neuronal acetylcholine receptors, right? So this is the basic classification of nicotinic receptor. So these are named based on the location they were initially found. Okay, so as the name suggests, muscle type acetylcholine receptor, that means these are usually expressed in the skeletal muscles. Well, nicotinic receptors were initially found in the skeletal muscles, and they uh, and the researchers found it mainly involves in maintaining, you know somatic innervations. Okay, so for the movement of limbs and voluntary movements, that's why they termed it as nicotinic muscle type receptors. Okay, so basically if I have to draw the central nervous system, and suppose some nerve innervations are reaching the skeletal muscle. Okay, at this particular region on the surface of the skeletal muscle, we have NN receptors. Whereas for the nicotinic neuronal receptors, as we talked about, pre and post junctional acetylcholine receptors, right? So suppose this is the pre-junctional, it is ending at a ganglia, right? And we are having a post junctional acetylcholine receptors, and that is further reaching, suppose, the skeletal muscle. Okay, so here at the post synaptic region of the post ganglionic neurons. Okay, so here this is basically an N type of nicotinic receptors. So these are basically present in the post ganglionic nerve. Okay, whereas here on the skeletal muscle, basically these are an M type of nicotinic receptors. Now, as I told you that basically there are 5 different subunits of nicotinic acetylcholine receptors present, that combines together to form a complete uh, ion channel, right? But both NN and NM type has different types of subunits which combines together to form a pentameric ion channel. So basically, if we talk about NM receptor, okay, or the muscle type receptor, uh, it has been found that in the adults there are certain combinations of this these 5 subunits. Okay, whereas in the fetal state there are some different combinations present in this nicotinic acetylcholine receptors. Okay, so what has been found that in the adults, basically there are two alpha 1 subunits. Okay, so out of 5 there are two alpha 1 subunits, one beta 1 subunit, okay, one delta subunit, and one epsilon subunit present, right? So total it forms 5 subunits that forms one complete nicotinic acetylcholine receptor, right? So it is 2, 3, 4, and 5. Whereas in case of fetal one, uh, the alpha 1 subunit is same. There are two alpha 1 subunit, one beta 1 subunit, okay, one delta subunit, and instead of epsilon there is one gamma subunit present. Okay, so this is the difference that has been seen in case of pre-birth as well as in case of adult nicotinic acetylcholine receptors. Okay, just keep in mind the two alpha subunit, these two alpha subunits play a very important role in ligand binding. Okay, as let me tell you about this. Okay, so this subunit combination is fixed, so it's not usually changed much in case of this NM type of receptor. So in NM type of receptor, what happens is the combination formed between these four type of subunits, these are basically fixed. Okay, these are not changed much. Okay, so for example, if I draw the structure of nicotinic receptor again, okay, so this is the pentameric structure, and as I told you, it has two alpha 1 subunits. So suppose alpha 1 is present here and here. Okay, one beta 1 is there, one suppose delta is there, and one is epsilon or gamma, based on if you are adult or in fetal state. Okay, now what happens, always the agonist binding domain are present between the two subunits. Okay, so they are not the agonist doesn't only bind to the single subunit, they are basically bind in the intersection of the two subunit. And always for the case of NM receptor, even for the case of NN receptor as well, one alpha subunit must be present, okay, where the agonist will bind. So in this case, as you can see, there are two alpha subunits, so the ligand can bind to this region, this would be alpha beta. It can bind to this region, again alpha beta, to this region, basically this is alpha delta, to this region, we can call it as alpha epsilon or alpha gamma, but never to this region, because this has no alpha subunit present, right? So basically for NM type of receptor, the most important acetylcholine binding site or the nicotinic binding site are basically the alpha delta and alpha gamma or epsilon, right? So this one or this one, alpha delta or alpha gamma or alpha epsilon. And this alpha beta interactions are very less common, so those are very limited. Okay, so mainly the acetylcholine binding site are these three particular intersections of nicotinic receptors. Okay, but if you have to talk about the N type of receptor or the neuronal type of receptors, what happens is they only form two like the combination of two different subunits. So they are formed of alpha and beta subunits. Okay, now among these alpha and beta subunit, as we know, these only two subunits are not enough. The ion channel is of 5 subunits, so there has to be some repeated alpha or repeated beta subunits must be present in the pentameric ion channel, right? So basically alpha has some different family of subunits. So basically alpha has alpha 2 to 10 subunits, and beta has beta 2 to beta 4 different subunits. Okay, so they differ in the amino acid sequence. Okay, and it can form combinations among different subunits. Okay, so it can form many combinations between alpha and beta subunits, right? So as you can see, there are alpha 2 to alpha 10 subunit and beta 2 to 4 subunit. So any of the alpha can bind with any of the beta, and even alpha can only bind with its own alpha subunits to form homomers, right? So but whereas in case of nicotinic muscle receptor, what we found is combinations are very fixed, right? Okay, so next, if you have to see what are the important combinations of N receptors present, okay, basically the most important combinations present or expressed in different organs, like in CNS or in neurons, are basically alpha 4 beta 2 and alpha 3 beta 4. These combinations are usually uh, present in the nervous system, okay. So what does alpha 4 beta 2 means? So suppose I draw a typical structure of nicotinic receptor again, okay. Now in this case, the combination will be between alpha 4 and beta 2 only. Suppose 3 are alpha 4 and 2 are beta 2, okay. So like this, it will form a combination between alpha 4 and beta 2 to form heteromeric, right? Heteromeric N receptor. Why heteromeric? Because it has alpha and beta subunits present. Now for example, if I draw a nicotinic receptor, okay, which has like alpha 2, alpha 4, beta 2, alpha 8, beta 4. Now uh, this has different combinations of alpha and beta subunit. Now the difference is in their nomenclature, okay. So basically now in this case it has only alpha 4 and beta 2 subunit, so we will basically write it as alpha 4 beta 2, right? But in this case we have to mention all the subunits as per the ascending order. So it will be alpha 2, alpha 4, alpha 8, beta 2, beta 4 nicotinic acetylcholine receptor. Is it clear to you? So the nomenclature is based on the alpha subunit first, and you have to arrange it as per the ascending order, and then the beta subunit as per the ascending order, and you have to name all the different subunits that are present in the particular ion channel. Okay, so this was all about heteromeric nicotinic receptors, right? Now in case of homomeric nicotinic receptors, what happens is, suppose we have the 5 subunit, so the subunit is always same, it is always 5 subunits. And suppose we have alpha 7, okay. So alpha 7 will be present in all the 5 subunits, so it will be alpha 7 nicotinic acetylcholine receptors, just a simple name, right? And this is obviously homomeric N receptor. Homomeric nicotinic receptors are not very common in case of NM, but NN usually forms homomeric nicotinic receptors as well as heteromeric receptors. Okay, so this was all about the different types of nicotinic receptors and how they are combined, what are the different subunits they can form, and how they basically works. Okay, now if I have to discuss about one single subunit of the nicotinic receptor. Okay, so as I told you, the complete nicotinic receptor forms of 5 different subunits. Okay, so if I discuss about only one subunit of the nicotinic receptor, let it be alpha, any of the alpha or any of the beta subunit. Okay, so the basic structure of one single subunit is it has 4 different transmembrane domains. Okay, so suppose this is the basic structure, and it has so this is the cell membrane, right? This is the cell membrane, and it has 4 different transmembrane domains. Okay, they are named as M1, M2, M3, and M4. Okay, now it has a long N terminal domain. Okay, I need some space here. So basically it has a very long N terminal domain, and N terminal domain has 2 certain amino acid residues. Okay, so let me draw in a different color. Okay, so it has 2 cysteine residues present at certain region in the N terminal, and they are formed they're basically separated by 13 amino acid distance. Okay, so they are basically separated by 13 amino acid different, so they are 13 amino acid far from each other. And what happens is the cysteine is basically sulfur containing amino acid, right? So if two cysteine molecules come very close to each other, they usually forms a disulfide bond. Okay, so as this n terminal loop is bit flexible in nature, and the two cysteine molecules are present just 13 amino acid apart from each other, they usually form a coil. Okay, they form a loop, and the two sulfur group of the cysteine molecules forms a disulfide bond, right? So that's why it basically forms a loop. Okay, so this, as you can see, this is a type of loop formation, and this particular loop formation is known as cysteine loop. Okay, or CYS loop. Now this is very important, as this is the basically a common structure or a reserved structure present in different ion channels, and there is a complete family of ion channels which has this common cysteine loop formation. For example, uh, the cysteine loop formation takes place in GABA receptors or GABA ion channels. It also it is also present in the glycine ion channels, right? So if the scientists see any ion channel that is forming this disulfide bond of the two cysteine molecules, which are separated from 13 amino acid apart from each other, they put that particular type of ion channel into cysteine loop family. Is that clear? So based on this, we identify this nicotinic acetylcholine receptor as a cysteine loop family ion channel, right? So, so this is the basic structure of a single subunit, right? Single subunit. And obviously the four transmembrane regions are also connected to each other like this. So these are the linker molecules, and the C-terminal domain is also extracellular. So the N and C-terminal both are extracellular in nature, right? So uh, yeah, I forgot to mention you about the thing is this cysteine loop. What is the significance of this disulfide bond? So it basically helps to stabilize the ligand binding domain. Okay, the ligand basically binds to this particular region of the N terminal. Okay, and it basically allows a proper room for the ligand to bind. Okay, so this was all about a single subunit of nicotinic acetylcholine receptors. Now the question is, where does the agonist or any drug binds to this particular receptor? As I told you, they basically bind to the extracellular region in the N-terminal domain here, but it is not that simple. Okay, so if we see the nicotinic receptor as a pentameric structure, okay, so as I told you that mainly the acetylcholine binding sites are alpha delta or alpha gamma region, like here, as I told you about. It could be alpha delta or alpha gamma or alpha epsilon region. Okay, so what happens is basically if we draw this particular one single subunit structure two times. Okay, so suppose, okay, so this is one subunit. Okay, and this is suppose another subunit, right? Now suppose this is the M1 region and this is the M1 region of the another subunit, and as I told you, we have a long N-terminal chain present here. Similar chain is present in this region as well. Okay, so suppose the two extracellular region are present close to each other. Okay, and they form a particular space for the agonist to bind in this particular region. Suppose this is alpha subunit and this is beta subunit. Okay, so it makes a particular conformation such that it basically forms a particular room or a cushion for the agonist to bind. So the agonist basically comes and binds to this particular region of the alpha and beta subunit of nicotinic receptors, right? So suppose acetylcholine or nicotine or any other drug, succinylcholine, etc. Those drugs can bind to this particular extracellular region and can open the channel. Okay, and obviously to show the effect, acetylcholine or nicotine has to bind to two different regions of the pentameric ion channel. Okay, so uh, suppose it is bound to one of the alpha delta subunit, and it is bound to the one of the alpha gamma subunit. So two molecules of the agonist must have to be bind to two different subunit, okay, to different region of the nicotinic receptor in order to completely open the ion channel. Okay, so that's an important factor that you have to keep it in mind, that always two molecules of the agonist is required to completely open the ion channel, right? So now the question is, how does the ion channel open? Now many of you will think that maybe the agonist will bind and that can cause some conformational change in order to open the ion channel, right? But it is not that easy. There is a particular arrangement that happens whenever a ligand binds to the particular region of the ion channels, okay? And one more thing that I want to mention here, that basically this M2 region forms the pore area of the ion channel, okay? So as you saw, this particular pore, right, this particular region from where the ions passes, this particular pore is being formed by the M2 subunit of alpha or beta subunit, okay? So basically what happens is, suppose we have 1 2 3 4 and here 5, right? So suppose this is M2, this is M2, this is M2, and this is M2. So all the four M2 will come together to form a particular pore, and from here only different ions will pass, mainly sodium ion, right? So M2 subunit basically forms the pore. Now, what basically happens? How uh, the channels are opened upon binding of the acetylcholine? Okay, so we can write. So the how this particular nicotinic acetylcholine receptor ion channel opens. Okay, so if we see, if we again make a diagram of nicotinic acetylcholine receptor, so this is the pentameric ion channel. Okay, okay, I have to keep some space on the top portion. Okay, so suppose this is the pentameric ion channel, and we have the N-terminal long loop, right? So all the alpha and beta subunit will have their own N-terminal loop. And suppose in the two regions, suppose in one region here and in another region here between alpha and beta subunit, two molecules of acetylcholine bound here. Okay, so when the acetylcholine bind to this particular region, suppose acetylcholine binding. So when the acetylcholine binds, what happens is it creates a conformational change in the extracellular region. Now what happens? The entire extracellular region twists or rotates a bit, like not that much, but a very less degree rotation takes place in the complete extracellular region. So this is these are the transmembrane regions, right? So when the acetylcholine binds to the particular ligand binding regions, now what happens? We find a particular orientation of the extracellular N-terminal region. Okay, now this particular thing takes place. Obviously the ligand is still bound to the ion channel, and so initially, as you can see, the N-terminal regions are a bit long and coiled, but now in this case there is a complete change in the conformation that took place. So the N-terminal or the extracellular region rotates or twists a bit. Okay, okay, so when the extracellular region twists a bit, it causes okay, so basically what happens in normal state, or we can say in uh, resting state, or when the channel is closed, okay, what happens is, as I told you, the M2 region of the of different subunits, right? They form the pore. Okay. Now the M2 region has some cluster of hydrophobic amino acids. So suppose if I draw the M2 region of 5 different subunits, right? So these are the 5 different M2 regions of the alpha and beta subunit. Now they have certain amino acids which are projected, which are hydrophobic in nature, and they projects like this. Okay. So all of them has some hydrophobic regions. Okay, so they have some hydrophobic regions, and they completely occlude the pore of the ion channel. Okay, so under the normal state, when the channel is closed, okay, when the channel is closed, there are some certain hydrophobic amino acid that causes that are present in a type of conformation which completely occludes the ion channel. So the ions cannot pass through this, because ion needs hydration in order to cross the cell membrane, right? So at closed state, this is basically taking place. But when the acetylcholine binds to the particular region and there is a twist of the extracellular region takes place, what happens is, now in this case, the M2 region of the uh, the alpha and beta subunit, they also rotates a bit. Okay, now there is a very slight rotation in the M2 region that takes place, but that slight rotation is enough to you know also rotate the hydrophobic regions. So the hydrophobic amino acids, they they come in a different conformation, so that it moves away from the pore region, and now the pore is completely empty, and there is no hydrophobic region present that can occlude the pore. Okay, so now the ions which are ready to cross the cell membrane can easily cross. Okay, so now the ions can easily cross and can enter the cell, basically sodium and calcium ions, and there could be some efflux of potassium ion that can also take place. Okay, so as the hydrophobic region shifted, okay, as the hydrophobic region shifted, it caused the entry of water molecules, or aqueous molecule here into the pore, and that also moved different cations into the cell or even outside the cell. So this is the basic mechanism of how the nicotinic acetylcholine receptor becomes activated upon binding of different agonists. Most important one is the acetylcholine and also nicotine, right? Now next we have to see, suppose the nicotinic acetylcholine receptor is open, that caused influx of cations into the cell. Now what will happen when the cations come inside the cell? Okay, so we have to see uh, the further effect after the activation of nicotinic receptors. So suppose these are the nicotinic ion channel, okay, and it is open because it got two acetylcholine molecules, suppose, binding at the two different regions. Now, uh, this nicotinic acetylcholine receptor caused influx of mainly sodium ion into the cell. Okay, now this sodium ion will initially, increase the positivity of the cell. So suppose we are talking about skeletal muscle. Okay, and suppose, for example, it has the resting membrane of around minus 70, or suppose minus 60 millivolt, right? So uh, or suppose minus 80 millivolts, because for smooth muscle it is basically minus 60, and skeletal muscle has bit negative resting membrane potentials. So suppose it has minus 80 millivolt of resting membrane potential, and when nicotinic acetylcholine receptor are active and it causes huge influx of sodium ion into the cell, the resting membrane, this resting membrane potential will start to rise. So it will start to become more positive. Okay, and suppose it reached to minus 60, or even minus 50 millivolt, right? So we can see a bit of a phenomena that you might know, that is known as depolarization. Okay, so as more sodium ion will enter the cell, the potential of the cell will start to rise. Okay, from minus 80, that is the resting membrane, it will start to go minus 60, minus 50, and at a particular time point it will reach a particular threshold at which the action potential is evoked. So suppose what happens if we draw a graph here, and suppose as more sodium is coming into the skeletal muscle, there is a rise in the membrane potential. Okay, so from suppose minus 80, it reached to, suppose, here minus 60, it went a bit up, and it reached a particular threshold, that threshold where more cationic channels are activated, suppose uh, minus 30 millivolt. Okay, this is just for example I'm talking about. So suppose at minus 30 certain cation channels are activated. Okay, at that case what will happen? Suppose it reached minus 30 millivolt, right? And in this case, what will happen, there are certain type of, suppose, cationic influx channel, for example, some voltage gated calcium channels. Okay, as these are voltage gated, so they will be activated upon depolarization. So suppose a particular threshold has been attained that can cause activation of more voltage gated calcium channel, so it will cause huge influx of calcium into the cell. So apart from the sodium, now we have huge calcium into the cell, and that will lead to huge increase in the in the membrane potential, and that can cause skeletal muscle contraction. Okay, and further it can go down by the balancing of the ions. Okay, so here we can see contraction. So this whole phenomena of binding of the agonist, opening of the nicotinic acetylcholine receptor to cause huge influx of sodium ion, and that sodium ion can cause increase in the membrane potential to cause depolarization. Okay, this whole process is known as end plate potential. Okay, because this particular phenomena usually happens in the skeletal muscle end plate. That's why this whole phenomena is known as end plate potential, or EPP. Okay, this is a very important phenomena that you might get questions on this in your examinations. So just keep it in mind that this particular EPP takes place in the skeletal muscle. Okay, and in most of the cases, this particular EPP leads to activation of more voltage-gated cation channels, or calcium channels that can cause skeletal muscle contractions. So they basically lead to large depolarization, right? And we need large depolarization to move to cause contraction in the skeletal muscle in order to move our limbs, right? So this is like very much important. And suppose if more voltage gated calcium channels are open, obviously the membrane potential will reach to, suppose here, -10, right? Even it can go to 0 millivolt as well. Okay, but ultimately it diminishes again and comes back to the resting state. Okay, so this was all about end plate potential. And one last thing is, so as I told you, this mainly happens in case of skeletal muscles, and we know in skeletal muscle we have basically n m type of nicotinic receptors. So what happens in neurons, so you have to know that as well. So basically what happens in neurons, and we know neurons are basically an n type of nicotinic receptors, and nn are located, as I told you, basically in the uh, ganglionic nerves, okay, or the postsynaptic regions. Okay, so suppose there is a release of acetylcholine that takes place from the preganglionic neurons, and here suppose there are some NN neurons present, right? Or NN receptors are present. Okay, so upon binding of the acetylcholine to this NN regions, what will happen, this NN receptor will be open, and they will cause certain depolarization again. Okay, so it is basically same as what we saw with an M type of receptor. In this case as well, when acetylcholine binds to the NN receptor, it causes depolarization, but the basic thing is this depolarization is very small. Okay, so it is a small depolarization which increases the potential to around 15 to 20 millivolts. Okay, so suppose initially there was a -65 millivolt at resting membrane. Okay, now neurons has a different resting membrane potential than skeletal muscles. We will talk about the resting membrane potentials later in a separate video. Okay, so suppose neurons has -65 millivolt resting membrane potential, and opening of an N type of nicotinic receptor will increase or make the membrane potential bit positive to, suppose, around -45 millivolt. So around 20 millivolt increase in the membrane potential takes place. This is not that much to cause contractions, and this is completely valid, right? Because nerves do not need to contract, they just need to transfer the action potential. And this much, I mean, the 20 millivolt rise in the membrane potential is enough to propagate action potential, and hence the nerve can completely work and release acetylcholine or even noradrenaline to the particular effector organs. Okay, so this is what you have to know about uh, NM and NN type of nicotinic receptors. And yeah, obviously this particular phenomena is known as EPSP, excitatory postsynaptic potential. Okay, EPSP, right? So this is the excitatory postsynaptic potential, Obviously, this is present in the postsynaptic neuron or the postganglionic neurons, and obviously, in nicotinic receptors are excitatory in nature. So this particular phenomenon is known as excitatory postsynaptic neurons, right? So this was all about two very important terminologies. One is the EPP, end plate potential, and other is the excitatory postsynaptic potential, or EPSP.
So till now, as we have discussed about the expression or the localization of different type of nicotinic receptors, which are mainly in the neuromuscular junction or in the postsynaptic neurons, right? So the all these expressional studies were found long time ago, and textbook just keep on repeating the expression of these nicotinic receptor in those particular locations only. But modern researchers they have found uh, that the nicotinic receptors are not only expressed in those two locations, but they are located in different types of other types of cells as well. Like so, as we know, earlier studies, right? They have found that the nicotinic receptors are mainly expressed in the neuromuscular junction and skeletal muscles, right? Okay, so neuromuscular junction, skeletal muscles, and postsynaptic neurons, right? But modern studies have found that so they are widely expressed in the brain, like different areas of brain. For example, dopaminergic neuron. Okay, even they are expressed in uh, airway smooth muscle, many epithelia, even in some mucus producing cells, and also in many inflammatory cells. Okay, so let me show you some paper so that it will be more clear to you, and you can go through that paper where many researchers have found the localization of this nicotinic receptor in different organs as well. So the first paper I found is this one, uh, which was written by Khalafi et al. In the year 2023. So they found the expression of alpha 7 nicotinic acetylcholine receptor. So alpha 7, as we discussed, it is a homomer type of nicotinic receptor formed of 5 alpha 7 subunits, right? So they found the expression of this alpha 7 nicotinic receptor in human airway smooth muscles, and they also stated that cigarette smoking or vaping can directly activate this particular nicotinic receptor, and that can increase airway smooth muscle contraction in asthma and COPD. So ultimately, that will cause more bronchoconstriction, right? So you can go through this paper, and you might be able to know more about the expression of nicotinic receptor in airway smooth muscle. The same lab also published another paper. So this is a review article. Okay, so they also discussed about the expression of nicotinic receptor and the role of nicotinic receptors in airway diseases. So in this paper also you will be able to know about the harmful effect of smoking and vaping, how it activates the nicotinic receptors, and that can increase bronchoconstriction. So this paper was published in 2024. It's not that old, so you guys can have a look and read this paper. So the next paper that I found is this one, which was published in the year 2012. So they basically stated that so they also found the expression of alpha 7 nicotinic acetylcholine receptors in mucus producing cells. Okay, so they found that in inflammatory diseases, basically the expression of this alpha 7 nicotinic receptor increases in the mucus cell, and then the mucus cell undergoes hyperplasia, and further there is upregulation of MUC5AC, that is the mucin producing gene. So the upregulation of that gene takes place, and that can increase the mucus formation. And they also discussed about the ill effect of cigarette smoking or nicotine smoking in airways. So you can obviously go through this paper to know more about the expression of nicotinic receptor in mucous cells. Okay. The next paper is on the expression of nicotinic receptor in epithelia. So apart from smooth muscle and mucus-producing cells, some of the researchers have also found the expression of alpha-7 nicotinic receptor in the airway epithelia. So I'm talking all of this about airways just to focus on how harmful cigarette smoking is. So cigarette smoking can activate the nicotinic receptors, which are expressed in different types of cells, in smooth muscles, in mucus-producing cells, in epithelia, which can be very harmful in different aspects. And finally, this is the paper by Maclean et al., which was published a bit a long time ago in 1998. But they found the expression of nicotinic receptors in vascular endothelial cells. Okay, so now you know, like it's not only limited to neuronal cells or skeletal muscles, but nicotinic receptors are expressed in variety of other types of cells, and they can play a huge different type of roles based on the cells they are expressed in. Okay, so this is what I wanted to focus you on, just to keep you guys updated about the expression of these particular receptors and what importance they play by expressing in different organs. Okay, so that you can target the disease properly. So finally, we have to discuss about the drugs which acts on these particular nicotinic receptors. Okay, so there are wide varieties of agonist and antagonist that can work on this particular nicotinic acetylcholine receptors in order to elicit some response. Okay, so as we know, as I have made this diagram quite several times, so you guys might know what is this. So this is the typical pentameric structure of nicotinic acetylcholine receptors, right? And it has extracellular and terminal where basically the the agonist binds, right? So as I have discussed about the binding site of acetylcholine and nicotine, that is in between the alpha and gamma or alpha or delta or alpha and epsilon subunits. Okay, so basically nicotine or acetylcholine binds in this particular extracellular regions. Okay, so the first drug is obviously acetylcholine and nicotine. They bind there, and they are the full activator. So they activates the this particular nicotinic receptors, right? And one thing to note that nicotine is also present in cigarette smoking as well as in vaping. So what happens is some of these nicotinic receptors are expressed in the dopaminergic neurons in the brain, right? So when someone smokes or vapes, what happens? The nicotine that goes into the circulation, they can go to the brain and can activate the nicotinic receptors present in the dopaminergic neurons. Okay, so so suppose this is the dopaminergic neurons, right? And it expresses different nicotinic receptors on their surface. So when these nicotinic receptors are activated, they basically stimulate the dopaminergic neuron to release more dopamine. Okay, so when dopamine is released, we feel a symptom of happiness. So we get happy, we get relaxed. It is basically a reward center, right? So we feel like we achieved something, right? So this makes us to get that same feeling again and again. That's why it is very harmful. It causes dependency and it makes us feel to get that particular feeling several times. That's why smoking is very harmful, or even vaping is very harmful in this particular regards. So the point is basically nicotine binds to the extracellular region in the N terminal of the nicotinic receptors, right? So apart from acetylcholine and nicotine, which acts as a full agonist, we have some drugs like varenicline. Okay, now this drug has a special characteristic. This acts as a partial agonist of alpha 4 beta 2 receptors. Okay, so this acts as a partial agonist. Now these were the full agonist. Okay, so the varenicline acts as a partial agonist of basically alpha 4 beta 2 receptor, which are mainly expressed in the neurons, right? So what happens is these drugs like varenicline, as they act as a partial agonist, they basically stimulate the receptor very mildly, like they do not fully activate the receptor. Okay, so they basically stimulates the receptor mildly, and you know prevents the full nicotine activation, right? So suppose nicotine was supposed to bind at this particular area, but in this place, varenicline is bound. Okay, so nicotine cannot bind to that particular region. Whereas the varenicline is bound to that region, it will produce very minimal effect or minimal opening of nicotinic receptor. So the full agonist activity of nicotine won't be there, and that's why it kind of like reduces the craving of nicotine over a long period of time. Okay, so that's why this particular drug varenicline is used for smoking cessation or smoking craving. Okay, it reduces smoking craving. So basically, if anyone is addicted to smoking as a treatment or as a therapy, varenicline is also given to them. Okay. Now, apart from that, there are certain antagonists also present. Okay. So basically, they are called as competitive antagonist. Now, you know the meaning of antagonist, right? So they will bind to the receptor, but they won't have any effect. So they, bind to the same site where acetylcholine or nicotine binds, but they does not has any, does not cause the opening of the nicotinic receptor, and they acts as a competitive in nature. So they will compete with nicotine and then prevent the nicotine binding, and instead of nicotine, they themselves will bind to that particular space, and they won't allow the channel to open. Some of the drugs, which example rocuronium, vecuronium, or tuberculorin, they fall under this competitive antagonist drugs. Vecuronium and tuberculorin. Okay, so they are basically, as nicotinic acetylcholine receptor, these are excitatory in nature, so activation of them can cause skeletal muscle contractions, right? So if we inhibit that particular nicotinic receptor, so that will cause skeletal muscle relaxation, right? So these are basically used as a muscle relaxant during surgery or maybe intubation. Okay. Now, one interesting point about tuberculosis is in the earlier times, whenever a group of people from tribal area they used to hunt. Okay, so as they used bow and arrow, what they did at the tip of the arrow, they used to put some a plant alkaloid. Okay, and that plant alkaloid had this particular tuberculosis in it. Okay, so they suppose they had a bow and an arrow, and in the tip of the arrow they put a plant alkaloid, and the plant name was curare. Okay, Q-R-E. So this curare had D-tuberculorin. Okay, and they used this bow and arrow to hunt for any animals, and when this arrow entered into animal's body, it caused skeletal muscle relaxation. Okay, so it basically causes paralysis in those animals, so the animal cannot move and they can easily catch that particular animal. So uh, this tuberculosin is used from very old period of time, and nowadays it is used in medical research to cause skeletal muscle relaxation in many surgeries. Okay. And finally, I would like to discuss about another type of drug that is called depolarizing blocker. Now, the example of this drug is succinylcholine. Now, what happens is this particular drug also binds to the same region where nicotine or acetylcholine binds in the extracellular region. But what happens is this acts as a persistent activator of nicotinic receptor. So they activate the receptor persistently, and what happens is if you keep the receptor active for a very prolonged period of time, then that causes deactivation. Okay, so what it basically does is it activates the receptor persistently. Okay, so the prolonged activation can cause, or the prolonged depolarization can cause deactivation of the receptors. Okay. So if the receptors are deactivated, so now no more nicotinic receptors are there to cause excitation of the cells, and ultimately it will lead to less nicotinic acetylcholine receptor activity, and that can again cause the effect that we saw with antagonist, right? So the thing is same; they are directly blocking the nicotinic acetylcholine receptors, but in this case they are activating it so much that that is causing blocking or blockage of nicotinic acetylcholine receptors. Now, this it is not a direct blockage. This is indirect blockage. Okay, they are basically causing uh, deactivation of nicotinic acetylcholine receptor, right? And this causes paralysis or skeletal muscle relaxation. So basically, succinylcholine is also used as a paralytic agent. Okay, but the mechanism of action is a bit different. So this was all about the drugs involved in activating or even inhibiting the nicotinic acetylcholine receptors. I hope you enjoyed this topic, and from the next lecture part, we will talk about muscarinic receptors in details.